This video, I'm just going to go over a couple of the topics we spoke about in recitation yesterday, of them being Mendelian genetics and how to assort them. So here we're given an example problem. It says gray is dominant to ebony, and long wings are dominant to vestigial. So we're going to assume that we crossed all the parental individuals, and they're homozygous. So let's say we crossed an individual that is dominant, that is gray with long wings, to the individual that is ebony with vestigial wings. So let's say we had, and we're going to assume that they're homozygous. Okay, so let's just do a small Punnett square. Okay, so these are both homozygous dominant for this organism right here, for this individual. And this right here is both homozygous recessive. So the offspring are all going to come out heterozygous at both loci. So it's going to have a big G, a little g, a big L, a little l, and just a small Punnett square to kind of show it. If we had, for example, big G, little g, we'd end up with all heterozygous alleles at the specific gene. Likewise, that would be the same for the L. So let's say we crossed the F2 progeny. So the F2 is all heterozygous at both loci. So we would have a big G, a little g, a big L, and a little l crossed with a big G, a little g, a big L, and a little l. Now, we would perform the same Punnett square for each of the alleles, or for each of the genes. So here we have a Punnett square that we're going to cross for the G, and a Punnett square we're going to cross for the L gene. We're going to have a big G, a little g, a big G, and a little g. And if we do the cross, we get many possibilities. So for the genotypes, we have 1 out of 4 that are going to have homozygous dominant genotype. We're going to have 2 out of 4, which will have the heterozygous genotype, which will be big G and little g, but this can be simplified down to 1/2, but it's better to keep it as 2/4, and you'll see in a moment why. And we're going to have 1/4 of the progeny, or of this ratio, which are going to come out little g, little g. Now, the cross for the L gene, it will give us the same results, since it's also heterozygous, cross another heterozygous individual. Big L, a little l. So we're going to have 1 out of 4, big L, 2 out of 4, or 1/2, big L, little l, and 1 out of 4 with little l. Now, in order to see all the different genotypes that can be produced, what we're going to do is we're going to take the ratio for the homozygous dominant at the G allele, at the G gene, and we're going to multiply that ratio with all the different ratios at the L gene. So we have a 1/4 possibility to have big G. We're going to multiply that with the 1/4 possibility of having big L. And that will give us a 1 out of 16 possibility of having an individual that has a big G, big L genotype. We're going to multiply it with 2 out of 4 as well, and the 1 out of 4. And this is where it's easier to keep it written as unsimplified, 2 out of 4. Because if you did it as 1/2, you'd then have to multiply it to get to a total of 16, because that's how many, because that's how big the Punnett square would be, since we'd have 2 genes, so 4x4 Punnett square for one gene, another 4x4 Punnett square for another gene, multiplied together, we'd have 16 total squares that we'd have to fill out. So this right here, multiply this together, we would get 2 out of 16 to get big G, big L, little l. For the little l, it would be a 1 out of 16 ratio, or possibility to get big G, little l. Now simply we would do this for all of it, for the rest of these as well. So we would do it for this as well, and we would do it for this, for the homozygous recessive. And that would give us all the different numbers of genotypes that we could have. Now all the phenotypes may be similar due to the fact that it does have a dominant allele. Like for example, this and this, they're going to have the same genotype, the same phenotype, but the genotypes differ at this one allele. So if we were to do it for the rest of them, oh no. So if we were to do this for the rest of them, we would end up getting 2 out of 16, this. So if we count up all the different phenotypes that we have, if we're going to have a gray individual with long wings, a gray individual with vestigial wings, an ebony individual with long wings, an ebony individual with vestigial wings. We would see that we have these two genotypes that will produce ebony individual, a gray individual with long wings. This will produce, these two also will produce an individual with gray and long wings. All of these right here will not. So we have 1 out of 16. This right here, if we add it to 2 out of 16, we have 3 out of 16. 3 out of 16 plus 2 would give us 5 out of 16, and then we'd end up with 9 out of 16. The individuals which we crossed will have gray and long wings. Now, for gray and vestigial wings, we count that we have one individual here which will have gray and vestigial wings, and another one right here which will have gray and vestigial wings. And if we add them together, we'll get 3 out of 16. So we 3 out of 16 individuals will have gray and vestigial wings. For the ebony with long wings, if we count that out, we see that there's only 2 of them, which will give ebony and long wings. If we add the ratios together, it'd be 3 out of 16, so it would just be 3. And if we look for ebony and vestigial wings, we see there's only 1 out of 16, which means we'd have 1. And so the ratio would be 9 to 3 to 3 to 1. And this is typically how heterozygous crosses go. This is the inheritance pattern for heterozygous individuals at both loci. It's always going to be a 9 to 3 to 3 to 1. If it differs from that, then perhaps the genes are on the same chromosome and they are crossing over or something's happening. All right, on to the next topic, which is Mendelian genetics and biochemical pathways. So if you guys remember, biochemical pathways are essentially a pathway in which it's a group of, you have a precursor molecule, which then goes through enzyme-mediated reactions, which will then produce the final product. So we have a precursor, which will be converted by enzyme A to then a couple intermediates. To say this is molecule Y, which is then converted by enzyme B. It's converted to molecule Z. All right. So we have precursor. It's converted by enzyme A into an intermediate, and then this molecule Y, the intermediate, is then converted by another enzyme into the final product. That is essentially a biochemical pathway. Now we can combine this with the Mendelian genetics. So let's say, for example, we have a pathway for producing pigment. All right, so we have an individual that starts off as white. It's converted by an enzyme. Let's say this right here is enzyme A. I'm just gonna write an A, and that's converted into a yellow color, which is then converted by enzyme B into a black color. So white, in this case, would act as a precursor. Black would act as the final product in the pathway. Excuse me. Let's say we had a mutation in one of the in the genes that codes for enzyme A or enzyme B. So this allele gives us, we have a big A, and this allele works, and then we have a mutated allele, little a. Now, if both of these mutated alleles are present and they are inherited, then the enzyme will not work. So if an individual receives two little a alleles, that the gene that codes for the enzyme A will have a defect in it and it will not code, and it will result in an enzyme that is not functional. So white cannot be converted into yellow because there's a defect in enzyme A. Likewise, if a defect happened in enzyme B, in which there was a defective allele, we have the big allele, big B, and that results in a working enzyme, and we have a defected allele, little b, and that results in defect in the production of the enzyme, which results in a defected enzyme which cannot convert yellow into black. All right. So let's say we had an individual that is black. And remember, we're always going to assume that it's homozygous dominant or homozygous at each of the loci. So here we have an individual that's black. So we're going to assume that the genotype of it will be big A, big A, big B, big B. And then we're going to cross that with an individual that's yellow. So we're going to assume that this individual is little a, big B, big B. So, oh, never mind. It's going to be big A, little b, little b. So the reason this right here would be big A, is because we're going to assume that it's homozygous. And we know that to go from white to yellow, enzyme A needs to be working in order to convert this pigment into the other pigment. To go from yellow to black, we know that enzyme B needs to be working to convert the yellow pigment into the black pigment. We were able to convert, or for this organism, it was able to go from white to yellow, which means that enzyme A is working, but it was unable to go from yellow to black, meaning that there's a defect in enzyme B, or the gene that codes for enzyme B. So for that reason, it's going to be homozygous recessive, signifying that there's a defect in that gene, meaning that it has both of the defective alleles. Now, let's say we cross these individuals together. what we'd do is we'd make a Punnett square for each of the alleles or the genes. So for gene A, we're gonna make a cross, and we have an individual that is homozygous dominant. Both of them are homozygous dominant, so all of the F1 progeny are going to be homozygous dominant at the A allele. At enzyme B, or at gene B, we see that one of the individuals is homozygous dominant, the other is homozygous recessive. If we were going to do a Mendelian cross or Punnett square, we will come to find that all of them will turn out to be heterozygous. It has at least one dominant allele, which means that it's coding and it's functional and it can produce the enzyme. So all of them. So these right here are all the genotypes of each of the genes. If we put them together, all the individuals are going to come out big A, big B, little b. So 4 out of 4 will have this genotype. 4 out of 4 have this genotype. Multiply it together. If we multiply both of these ratios, it'd be 4 out of 4 times 4 out of 4, which will give us 16 out of 16, which means all of them will have this genotype. Now let's say we cross the F1. So we're going to cross the big A, big B, little b with another individual who's big A, big B, little b. So we're going to do the same. We're going to make a Punnett square for each of the genes. We're going to do a cross. So they're all big A. All are still going to come out. 4 out of 4 of them are going to come out. All of them are going to come out with the genotype big A at the A allele, at the A gene. Now, at the B gene, if we were to perform a cross, we know that they're heterozygous. And so we're going to have a couple different genotypes that are going to pop out. So we're going to have 1/4, which are going to have the genotype for big B. 2 out of 4, which are going to have the genotype for big B, little b. 1 out of 4, which are going to have the genotype for little b. So if we multiply all these together, as we did in the above example, we would get 4 out of 6. We would get 4 out of 16 of the individuals will have the genotype big A, big B. We would get 8 out of 16 of the individuals in the progeny which have big A, big B, little b, and we would get 4 out of 16 of the individuals in the progeny will have big A, little b. So these right here code for working enzymes, which can convert the yellow into the black. So these will have the black phenotype. These individuals have a defect in the enzyme B, and they have both of the defected alleles, which means they cannot convert yellow to black. So these individuals will turn out yellow. So the ratio of black to yellow would be 12 to 4. Or if you simplify that, it would go down to 3 to 1. All right, on to the next topic, which is going to be chi-square. So chi-square, what that essentially is chi-squared is a statistical test used to calculate differences between variables to see if they're independent of one another. Or in other words, see, if they're independent of one another, or if one is impacting the other, or if there's a relationship between both of them. Now you're going to be given things known as such as the null hypothesis, and the null hypothesis essentially states that there's no significant difference between the observed and expected data, meaning that there's no relationship between the variables. So there's no significant difference between the observed and the expected data. And so you're going to end up with something, and you're going to have a p-value. So a p-value of less than 0.05 says that there's less than a 5% probability that the null hypothesis is correct. So that it says that there's a less than 5% probability that the results are random and not related, so that they are independent of one another. If you have a p-value less than that, that means that the null hypothesis, less than 0.05, that means the null hypothesis is incorrect. Rather, the variables are related somehow, or there's something strange going on between them. If the p-value is greater than 0.05, then you want to accept the null and say that the variables are independent of one another, and that there is no relation between them. And you also have things referred to as degrees of freedom, df. Or I think it's going to be written little d, little f. And what that essentially is, it's the number of independent pieces used to calculate the statistic. So for example, we have two phenotypes, right? We have two, so the df, to calculate df, it's n minus 1, in which n is the number of phenotypic crosses, or phenotypic domains. So for example, we have two variables. Let's say that one of the variables is about, let's say hair color. It's brown versus white. There are two phenotypic classes, so the df for that would be 2 minus 1, which would equal 1. So df would equal 1. Well, I had to cut this video out really quickly in order to make an example problem because I didn't want to make this video too long. So let's say, for example, we had a total progeny of an organism, let's say deer, and 905 of them had antlers, and 95 of them did not have antlers. So the total came out to 1,000. What we would do is we would calculate the expected ratios. Let's say the expected ratio is 3/4. 3/4 for having antlers and 1/4 for having no antlers. The expected ratio for antlers would be 3 out of 4. No antlers, 1 out of 4. We saw and we observed 905 of them that had antlers, and we saw 95 of them which did not have antlers. The expected ratio, the expected amount of individuals without antlers or with antlers would simply be the total number of the progeny multiplied by the expected ratio. The expected ratio is 3 out of 4 or 0.75. Multiply that by 1,000. So we would expect 750 of the individuals to have antlers. The expected ratio, the expected individuals with no antlers would simply be 1 out of 4 or 25%. Multiply that by 1,000 and we'd get 250 individuals who we would have expected to not have any antlers. So then we would need to find the deviation, which is simply observed minus expected. So that would be 905 minus 750, would be 155. For no antlers, it would be 95 minus 250, which would be a negative 155. We would then do the deviation squared. So we'd simply square our answer from the deviation, which would give us 24,025 for both of them. And then in order to find the x-squared value, and the x-squared value is it's essentially the value which tells you whether or not it's the method. So the x-squared is the measure of difference between the observed and the expected frequencies. So it's the measure of difference between the observed and the expected frequencies. So we would calculate the x-squared value for both of them. We would take observed minus expected squared divided by the expected. We would add both the values to get the x-squared value. And now what we would do is we would go to the chart and we would check at 1 degree of freedom the x-squared value to see the p-value that is associated with it. So we see that right here at 1 degree of freedom we have an x-squared value of 128. So 0.05, if the p-value is greater than 0.05, then we're going to accept the null hypothesis. If it's less than 0.05, we're going to reject the null hypothesis. So we can see at 1 degree of freedom, we have 128, that's our value, 128. We see at 1 degree freedom, further away, or the larger the p-value gets, the smaller the x-squared value gets. And the further towards the larger x-squared value, the smaller the p-value gets. So we can see right here at 0.05 it's 3.84, 0.025 as it's getting smaller it's 5.02. So this x-squared value would be somewhere all the way down here. So we know that the p-value associated for it would be smaller than 0.05, which means that we would reject the null hypothesis. On to the final topic, which is Hardy-Weinberg. So Hardy-Weinberg equilibrium, that's essentially just it's a method to calculate the genetic variation of a population at equilibrium. At equilibrium, when a population is at equilibrium, that means that the population remains constant from generation to generation. So now there's 5 assumptions that are associated with the Hardy-Weinberg equilibrium. I'm not sure if you guys have to know them, but nevertheless, it's good to know them. The first of them being there's no mutations occurring. Second being that there's no gene flow, so there's no immigration or migration. Third being there's no genetic drift. And genetic drift is essentially a change in the allele frequency due to random chance, and that typically happens in small populations. So that would mean that the population is large. Assumption number 4 would be random mating is occurring. And assumption 5 would be that no natural selection is occurring. So if the assumptions are met, then 2 equations can be used. One equation can be used to determine the genotype frequency. So that would be p squared plus 2pq plus q squared equals 1, where p squared is the percentage of homozygous dominant individuals in the population, 2pq is the percentage of heterozygous individuals in the population, and q squared is the percentage of homozygous recessive individuals in the population, and that all would add up to 1. There's one more equation, and that is the allele frequency equation. And that equation would simply be p plus q equals 1. And remember, these all have to be in percentages. So if you're, for example, given a problem and it said that 21 individuals in the total population of 100 carry the recessive phenotype. In order to get this into a percentage, to find q squared, you would simply take 21 divided by 100, and that would give you something like, I think it just 0.21, if I'm not mistaken. Now, onto the problem. We're going to do part A. So consider a rare disorder in a population caused by an autosomal recessive mutation. From the frequencies of the disorder in the population given, calculate the percentage of heterozygous carriers. Okay, so we're given the frequency of this order in the population, which means that we are given and of the recessive mutation, which means that we were given q squared. So q squared equals 0.0064. So they want us to find the amount of heterozygous carriers in the population. So we would have to take q squared, find the allele frequencies, and then plug those allele frequencies into pq in order to get the amount of the frequency of heterozygous individuals in the population. So q squared equals 0.064. In order to get q, we would simply square root it, which would then give us q equaling 0.08. And so the equation for the allele frequency is p plus q equals 1. Q is 0.08 equals 1, which would mean that p equals 0.92. In order to find the heterozygous, it'd be the equation that we're going to plug it into is just 2pq. So 2 times 0.92 times 0.08. And that would give us 0.1472, or 14%. And if you want to check over your answer, what you can do is you simply plug p and q into the genotype frequency equation. So it'd be 0.92 squared, and you'd see if it all equals out to 1. 0.92 squared plus 2 0.92 times 0.08 times 0.08 squared, and you see if this equals out to 1. In this case, it does equal out to 1, so the answer is correct, and so it would be 14%.